first lecture session of First Stage STTP 2021. Today, we are fortunate to have with us Dr. Balram Ghosh as our guest speaker for this session. Dr. Balram Ghosh completed his B.Pharm and M.Pharm from Jadavpur University, Kolkata. He was awarded with the PhD degree from Department of Pharmaceutical Science, Wayne State University, Michigan, USA, and he was postdoctoral research fellow at Harvard Medical School, Boston, USA. Sir has more than 20 years of teaching and research experience. To his credit, Sir has been awarded with Frank O. Taylor Best Graduate Student 2008 Award for outstanding performance in research and graduate studies, Wayne State University, USA. Adding to his credit, Sir has more than 790 publications in various national and international journals of repute. His major research interest area, development of subtype specific histone deacylase inhibitor activator as epigenetic modulators, mainly potent and isoform selective HDAC inhibitors activators to understand the individual isoforms of HDACs responsible to turn on and turn off the transcription mechanism involved in with the activity candidate genes. Presently, Sir holds the position of Assistant Professor, Department of Pharmacy, BITS Pilani, Hyderabad campus. We welcome you, Sir. Now, may I request Sir to start with his deliberation, please, Sir?
Thank you very I'm very privileged. Can you hear me?
Yes, Sir, you are audible.
Yeah, yes. So I'm very privileged to get this platform to talk about a few things, part of my research. I'm very thankful to the to Columbia Institute of Columbian Group of Columbia Institutes, especially Professor Amit Roy and Sanjeev Bahadur, and may all of you, the organizing committee. Actually, today's talk, even though it is a some teaching learning program focused, so I will start with a few things related to teaching learning. Then I'll go to my own domain of research, and I'll not complicate. I'll not present in the complicated way of research, but I'll try to present in a way that people, those who are not in this field, can understand or can at least realize what is happening. So with your permission, I'm going to present it.
Sure, Sir.
So it's my apology that I'm not very well prepared for the today's talk, as we are overburdened with lot of evaluation component going on in our institute. I just made few slides. It will be better. It will be very helpful for me. It's my earnest request to you all. Please make it interactive. You stop me anytime. We can discuss if something interests you. So today, my research talk title is Learning and Memory. Yes, we can enhance it. Why I said yes, we can enhance it, because might be there is a notion or impression that with learning and memory, it's inherited property of a human being, animal human being. It cannot be enhanced, or it is not decreased. I was also under impression quite some time ago, before I come to this research, that we all born with the same capability of learning and memory. It depends upon how much we study, how much we practice. But at some point, I understood that it's not. It's not. You can enhance it, or some kids, they are born with the compromised learning and memory. Learning and memory, as I said, I want to say it's a very, very complicated procedure, and it involves many things, many neurotransmitter, many interactions between the neurons. There are different types of learning and memory. I'll just give some of the definitions of different types of learning and memory, and then I'll go further. Learning, if I explain what is learning, you see, in old days, Guruji was there, and whatever he used to say, that was the only way of learning. You listen it, memorize it, use it. Then it replaced by the hard copies. You have textbooks, you have many hard copies in the textbooks or printed copies of it. Just go through it, memorize it, understand it, apply it. In the 20th century, the learning was mostly .com era. Means whatever you want to learn, www.this.com. You put it in the internet, you'll get it. It was not, you don't have to buy a textbook, you don't have to buy a printed copy. Many textbooks are available. Software, mostly all of them are available. Some of them are paid, some of them are free. But the 21st century, I would say it's a click era. Learning is a click era. You just want to click. You don't want to download. You don't want to put that www.something.com. You don't want to do that because this science has advanced in a huge amount, in a huge way. You just click it. So click era. What I mean by click era? You see, I attended a few lectures that is being organized by you people. So some big people who are there in this field for a long time. Some people say, no, don't keep the text in the presentation. You just keep the picture. You just keep the figures. I would say the text is also important, but keywords, and make them hyperlinked. You know hyperlinked? Hyperlink means, suppose neuron. You keep it here, it's a hyperlinked. You can link this one to the source where you want to explain it. Wikipedia, some paper, some journal, PubMed. So hyperlink this. Because nowadays we have the mobile phone. That is the most usable source or way of learning we are having today. We don't want to type the www.com. We just bring our cursor there. It will be hyperlinked. It will take you all the way to the link to that site, and it will be the definition. It's working here. Yeah. So they are all hyperlinked. See, it's coming. Page is loading, and you see Wikipedia came. These are hyperlinked. So every keyword can be hyperlinked to that. Okay. So then if the student is interested, he can go through a little bit of it, or say, oh, it's too boring. I need only the what is neuron. So this era is a hyperlinked era. In the mobile, you just see the text and the PowerPoint presentation teacher have shared. So just it will have few keywords, and you can just click it, you will get all the details. So actually that will bring interest in the students. Instead of giving lot of text, lot of picture, lot of figure, just use the hyperlinked keywords to define anything, to make them understand anything, to do anything. They will click it, they will go back to the original source where you got it. But make sure what you are hyperlinking, it has enough information there in the linked website. So I would say today is the main learning tool. Today's main learning tool is mobile with click option, and that will give the interest. Students, they come to the class, it's fine. If for us, we don't have the attendance compulsory. We share the slides even before they come to the class. So yeah, class is also important. I will come to that point why coming to the class is also important. But students might be smart enough. They think that they can manage with this click option of the presentation or topic you are covering. That is what I wanted to say about the way of teaching. So how we can enrich our students, how we can develop the interest into the students. That is, I think, to me, that is one of the best options to share the text, share the PowerPoint present slides, and minimum words with the hyperlinked option. Regarding motivation to do research, or thinking what to do research, what to do, how to do all these things, I'll start with this advertisement. You see, there is Horlicks. It is available with us. Get taller, stronger, sharper. You buy Horlicks. You have Complan, yeah, faster growth. You have Granola, that healthy memory. So these are the things we have it. It always struck in my mind, because when I was studying in 10 plus 2 grade, the syllabus was huge, and I was thinking, how can I increase my memory? I'm forgetting every day. So it was actually, I had a lot of things to ask that time. Can we not increase our memory so that I can read a book and I can keep in memorize for at least for 6 months, for 1 year, up to at least till the exam comes. Actually, there was nothing, and today also there are some things, but I would say it's also nothing. It's emerging field. It's just beginning field of memory. And the composition of the Horlicks Complan that we have, those we said for the kids that, Baba, you have to eat this, you have to drink that, you will increase your memory, you will be strong, your mental growth will be faster. A lot of ads we see that with the Complan, Bonvita, Horlicks, memory got increased. But is there something we have that can increase the cognition or learning and memory? So before we ask the question, then we have to define what is the cognition means, what is learning, what is memory. At least few little information we need to have it before we asking the question. So cognition is a process of acquiring and understanding the knowledge through people's thought, experience, sense. That is a cognition. And this cognition main key or main component is the memorization. So memorization is the one of the most important part for cognition. You will be brilliant, you will be intelligent. Even though sometimes we get the example, the Einstein had little memory, we could say that as least as possible memory he had, but he was the most brilliant, intelligent so far. Even he was going by train to somewhere, and then he had the ticket in her pocket, in his pocket, and the ticket collector asked, "Where is the ticket?" Then he was searching for the ticket, but fortunately the ticket collector recognized his Einstein, and he said, okay sir, you don't have to find it, I know you are Einstein." But then he left Einstein, went to some other passenger and came back, and again he saw that Einstein is still searching his ticket. Then the ticket collector said, "Sir, I said I don't need to see the ticket." The Einstein said, No, I'm not searching the ticket to show you, but I'm searching the ticket to know where I'll be going down, where I will go down, where I'm going to go. See how little memory he has it. Question is not that, or that is not the real story. Real story is he was engaged in his research, in his work so much that he had no space available for other thing to keep. Today we can explain better way that if you have a pen drive or external drive, your memory is full, capacity is full, you cannot put it more. So he made himself in such a way that all his memory was full. It was filled up with his own work, so that he even forgot, he did not have minimum space available to keep in mind or to memorize or store. I would say it's a storing event, phenomena. So to keep the where he is going to go by the train. So memory. So people sometimes memory. The people talk about. I know you don't need to memorize it. Just understand it. No need to read more. No need to memorize everything. Just try to understand. Try to answer the how as much as you can from your own. What is from your own? No, there is no your own. It is all about what you have stored in your brain. I'll come to that. So learning is a process of acquiring new information, and memory is the ability to store and retrieve. So if you have the file stored in your drive, it has to open it. It should be retrievable. So memory is also the same thing. You store it in a retrievable form, and those are stored in a specific area of the brain. The functional perspective of memory, the memory, there are several kinds of memory. I said I will not go in details on that, but at least few things. It's a short-term memory, then you have long-term memory, and then you have intermediate memory. Okay. So short-term, intermediate, and long-term. Three memories are there. So successful processes capture, store, retrieve information. They are stored in different brain regions. So this long-term memory actually the most useful memory. Short-term memory, yeah, it is useful for everything you do every day, but actually long-term memory make you more smart, more intelligent. You are brilliant. So there are again two types: declarative and non-declarative. Declarative means things you know that you can tell to others, and non-declarative things that you know you can show by it to others by doing it. Then again, there are different parts. I'm not going in details of that. So then short-term memory usually stands for few seconds, and then it's a working memory. Here it comes. That study we do every day, it's basically a short-term memory. As I said, it's a few seconds, right? But the short-term memory can sustain for a long period of time if you make it rehearsed. Means you are using it every time, you are memorizing, you are keeping, you are nurturing it every time, every second, every minute. So it stays for until the end of exam. So it's a short-term memory. You are rehearsing it till the exam. Then it is again short-term memory are of three types: it's audio information, visual information, and integrated of audio and visual. So as I said that many students say that my memory is very bad. I'm very poor in memorizing. See, you have to go in little detail of that before you say something. You are very poor. So the thing is that you have to nurture it. These are all short-term memory. You rehearsal, make it rehearsal, and make it long-term. At least working memory for a certain period of time, which is actually persist for few seconds. But nurturing or rehearsaling make them long-term. Then audio is shorter, visual is little longer than audio, and integrated of audio and visual is that can stay even further. Integrated long-term memory last for integrated term memory is a little more than the short-term, and long-term memory stays for years. So we'll be talking about the long-term memory. Long-term memory has the capacity to keep, you have a space in your brain. You have the storage capacity in your brain to keep the long-term memory. But it can be altered. How it can be altered? It can be altered or affected, meaning that your stored memory can be altered or affected by the new one or by the many other events before or after. So I was preparing for exam. I'm almost ready to go in the next morning, and in the night you have some accident in the family, in the neighbor. So your stored memory for the exam will be affected for sure. It's not your fault because it's a replaceable. Long-term memory is replaceable or changeable. So that's why I said this exam score was not good. Yes, my friend, that day before the exam, or just before my exam, there was incident happened. That's why my exam did not go well. But yeah, that is the explanation, that your long-term memory got affected by the incidents happened before the exam, so that you cannot retrieve it properly. Before you even retrieve it, they got changed, affected from the size. This file got virus affected. Some you can retrieve it, not all of it. But yeah, you can do all of it if you don't allow the accident to make a memory in your memory space. So this is a schematic quick diagram. These are the way of things by which your sensory buffers work. They come as a memory, encoding them. So it's a working memory first and short-term memory. Working memory, immediately you are performing. Short-term memory and working. Short-term memory needs consolidated to become a long-term memory. So every long-term memory starts with a short-term memory. The short-term memory, it consolidates and reconsolidates to become a long-term memory. That takes time. That consolidation is the important part. Sometimes I say that you are studying, but you are not memorizing, meaning you are not consolidating. You are not keeping these two all the way to the long-term storage place. So that's how it man to man, people to person, animal to animal, it varies. So strong emotion can make the consolidation better. Meaning that any incidents it happened, or anything you want to memorize, if add emotion into it. Sometimes these are inspiration, sometimes these are some personal relationship, sometimes your parents' affection. So you add emotion. So the short-term memory, when it will be consolidated to the long-term memory, it stores for longer period of time. So attachment with the emotion makes the memory stronger or longer. That is all about little part of the learning and memory. I'm not going the neuronal circuit. I'm not going the different brain region, hippocampus, cortex, nucleus accumbens, those parts, how the memory stored and consolidated and goes to the other part of the brain region. I'm not going into that. Just to give some idea, little idea about the memory and its consolidation, its storage, and working memory. There are some compounds already in the market. People are weak, or it is claimed that they are memory enhancer. And there are some biomolecules actually responsible for the memory formation. That means audio or visual to encoding them as a memory. So acetylcholine, one of them, epinephrine, non-epinephrine, vasopressin, opioids, and GABA. So these are the few biochemicals. They are involved in memory formation. There are some medicine also in the market that are actually working on these biomolecules that is involved in the memory formation. So there is a nootropics. There are a class of drug that says that they improve the memory, and these nootropics are memory enhancing supplements or medicine, has a huge market, because today we are fighting with each other to go faster, to be better, to be in a very competitive society. So this has a huge amount of market. And these are the chemicals or herbs or Ayurveda synthetic molecules. Those are in the dark market, used as a learning and memory enhancer. I will come to the limitation of these things. And these are the genes, many genes involved that upregulated or downregulated. Because of that, your memory is compromised. My question is, or when I started, I did not work in my PhD or postdoc on learning and memory. Here I started working on this. So when I dig into the literature, there was not very systematic study that where from it starts the problem of learning and memory. Is it the gene or is the epigenomes? Epigenome means gene sequence and nucleotide sequence is the same, but the product coming from the gene varies. Why it varies? Because from the gene to the product, there are many steps involved. And those steps, there are many catalysts, solvent, reagent, not the in the organic chemistry, but in the brain, in the human body. Like these are called transcripts or any enzymes, they are involved. So they don't do function, they don't do their function properly. That makes a difference. And sometimes it happens the gene is not performing well. They are repressed, they are suppressed. That's why memory got compromised. So my study started that what are the genes. When I was looking at the genes, if you put it in the PubMed, Google, you will see list hundreds of genes. So we just try to pick a few genes. Those are mostly affected or responsible for the compromised memory. And then we came into, I worked on the HDAC for a long time, and we saw that some people have done some work there. They have shown that this HDAC is one of the parameters or key player for better performance in the learning and memory. So learning and memory, they always come together. You have to learn, then you memorize, right? Without learning, memory doesn't exist. And learning, if it does not stay for at least few seconds, means if it is not memorized, the learning is not anything. So this HDAC is a histone deacetylase, is an enzyme which has a key role in the memory. So how it works, we saw that. So this is this picture. I'll put the pointer. So this is actually histone protein. It's an octamer, and DNA wrapped around the histones. So from DNA to RNA is the transcription. So when DNA works, it's the gene expression, right? When particular DNA part of the DNA is active, it's the gene expression. So if the DNA is tightly wrapped, then it is not capable of producing or doing any work. When they are tightly packed and when they are loosely packed, it depends upon the DNA. DNA is negatively charged. The amino acid residue of the histone protein, which contains lysine, is a positively charged group. So if the lysine is deacylated or in the only lysine form, then a lot of positive charge, and DNA is a negative charge, they make it very compact. Once they are compact, they are repressed form. They are not active form. If they are repressed, then your gene cannot work. Means you are not getting the product you are supposed to get from the DNA. So DNA is tightly packed, more tightly. So it is not relaxed. It is not transcribable. So we found from the literature that yes, if you inhibit that enzyme, histone deacetylase, then it will be acetylated form. And acetylated DNA actually transcribable. So we have to inhibit this enzyme, HDAC. So when we wanted to inhibit the HDAC, before that we saw who are the HDACs. There are many, 18 HDAC totals, 18 isoforms. Are all HDAC responsible for inhibiting the gene expression of learning and memory? There are a lot of literatures, they use the pan inhibitors, meaning that molecules that inhibit all the HDACs. They got the result. Some of them are class specific inhibitor, they got the result. There are few studies, very few studies. One is from University of California at Irvine, another from Liu Sai from MIT. They studied that HDAC3 has more important role in HDAC3 isoform of 18 HDAC role to play in the learning and memory genes. Since I had some idea or expertise how to make isoform specific histone deacetylase inhibitors or activators, I started making the HDAC3 histone deacetylase 3 inhibitors. How do we make it? This is a three-dimensional structure. It is available, protein structure available. Take many theoretical molecules. You have some idea that what kind of molecules will inhibit HDACs. Then make some theoretical molecules, simulate them in the active site of the enzyme, and find out the score. Do some virtual screening and come up with a list of molecules from the theory. Once you have a sizable, a working list of molecules, means you have manageable list. I had around 20 molecules. So I came up with the 20 molecules. Once I started screening virtually, means just drawing and simulation, 50,000 molecules, I came up with around 10 molecules that I decided to I'll make these molecules and validate the theory, whatever we are getting, that this would be inhibitor of HDAC3 or not. From that 20 molecule, we got one of the molecule most promising. Still, it is not 100% HDAC3 selective. They inhibit other HDAC also, but they have more potency towards HDAC3, 245 nanomolar. What these are 2 micromolar, 1 micromolar. Okay, so we came up with this molecule. Then once we got this molecule, I did not go in detail the design, synthesis of all this, but yeah, from the protein structure, crystal structure, simulation theory, we came up with the 20 molecules, and from the 20 molecules we made in our lab, it's a step-by-step multi-step synthesis, and we screened them for the enzyme inhibition for all of the HDACs. Then we found out that it has a potency towards HDAC3 is more. So that we named as PT3. PT actually stands for one of my students, Prakriti Trivedi. So it's my trend in my lab that whoever will make the molecule will give the name as per their name, of their initials. So the PT3 came up as a good molecule. Then see this HDAC inhibitors, when we are talking about the learning and memory, you have to find out this molecule has to go to brain. It has to be delivered in the brain. The problem is many times it happens when you do the computer-based drug design, then synthesize it, and you get to know that you got the molecule in vitro assay, in cell culture or enzyme-based assay, it works. But when you take it to the further in the whole animal, this in vivo, you don't get the activity. So the first thing you have to see that the molecule you are making, is it deliverable to that site of action? Where it will go and work? It has to go first there. No, you need to have you need to have the capability or potential to be reached there. So we first saw that how much is the drug distribution in the brain. So we did the pharmacokinetic study. Surprisingly, the right-hand side, you see, the left-hand side is the plasma concentration, and this one is brain concentration of the drug. You see, it is double the concentration in the brain. It is a serendipity. It never happens. You always get, you open the literature, you always see that molecules have more concentration in the plasma, and it has a substantial amount, required amount in the brain. But it never happens that it has double the concentration in the brain compared to plasma. You can say why it happens at then. Yeah, see, many things happen, then we try to explain. So if you see the structure, this actually, the pyrazine, we had the benzene version of it. The pyrazine version actually made the drug to be available more in the brain. That is explanation, hypothesis. I don't know exactly what is happening. But you see that the 1000 nanogram per ml, you see there's a more than 2000 nanogram per gram. It is more than what we have in the plasma. Then what we do, we design. Then we directly went to the memory experiment. There are many animal protocols to test the molecules in learning and memory. So there are freezing point. There is a shock-induced model. There is a object recognition model. There is a force swim test. There are many. So one of them, most acceptable long-term memory model is novel object recognition model. Just quickly, I'm trying to tell you that you have the cage, you keep the animal for habituation, take the animal out, keep the two objects here first, and you see that from first day to 10 days, 11 days, we are up to 10 days we are habituating. 1 day we are keep it for acquisition with the objects, and then on the 12th day, one of the objects, you remove it and put a novel object. Then you test the animal, how much time it is spending in exploring the novel object. So you see, you have the stored memory, but using that memory, you are exploring others. That's the cognition. Okay. So if you have the stored memory for strong means it's a more memory storage, then you will spend more time on the novel object. That correlation is there. Then on the 10th day we test it, 12th day we test it, and then we sacrifice, and we found out what are the genes. The 10 genes I said we picked from the list of genes. We just tested those 10 genes. That in the brain sample, we took the brain sample, homogenized it, isolated the cells, and then we did the RT-PCR, and we saw are those genes at all increased, gene activity is increased or less, or decreased or the normal. So I have a video of the test of the experiment. Might be it is interesting for you. So this is the test. This is the PT3. You see, you have two objects here, object 1 and 2 similar, and then you replace the object number 1 by a different object. It's a novel object, and then you are trying to see how much time is spent on the novel object recognition. And that time will be recorded from the camera, and you have to score it. That is scoring of that. So you see how much time is coming and exploring that novel object. So that is the parameter of determining the memory. Okay. So yeah, we have all of them. They are all different groups. Each group had 5 animals. Then each group has control, standard treated, non-standard treated. So the test treated all the parameters were there, and we saw that control animal, if it is it's a anonymous arbitrary axis, the time in it, and this much time, and then the test compound or PT3, you see how much memory. These mice are normal mice, wild type mice, and we tested all the genes that we said that from the literature we saw the many genes are involved, and we picked the best more involved or 10 most important genes, and we saw the genes are upregulated. Means after treatment of the HDAC, inhibitor PT3, we got those genes to be expressed to work more than the normal. And then we published that one in Nature Clinical Neuroscience. We did not do much work. We did very little amount of work as far as the publications are concerned. But I think the design part or the thinking of the experiment, total conceiving the idea and execution the idea, it got accepted. It will be published in 1 or 2 days. Then next, on the base on that, we got very excited. Then we started to see how further we can do it. Oh, that was for publication is fine. We got a good publication in HS Clinical Neuroscience, but what we can do next? Then we are searching that how we can do it related to the kids' memory and all these things. So we saw it. They have reported that pregnant mice, you give them normal diet and do their learning and memory test, you will get a data, a value. Then they have reported that pregnant mice with, it's not normal diet, it should be deficient diet. Yeah, it should be deficient diet. With the deficient diet you give, and then they will when the pups will come, the pups will have compromised memory. So that is already established in few literatures, that when the mother having nutritional deficiency, the pups are born with a compromised memory. So that's why it happens that poor children having compromised memory, learning and memory compared to the rich. This is a normal phenomena, but it's not that always true. There must be some exceptional. Even we saw that some kids are from the poor family are very exceptionally brilliant. But normal phenomena, if you have the mother has the malnutrition or nutritional deficiency during the pregnancy period, the pups will be born with the deficient of learning and memory. Then what we did, we planned an experiment protocol and submitted to DBT for the funding, and we got recommendation. This one very simple experiment. Take the pregnant mice, give the normal diet, get the pups, and do the learning and memory. You get a value. Then give the deficient diet, get the pups, do the learning and memory. As expected from the literature, you will get a compromised learning and memory. Our next, this is our question for the proposal. Take the pregnant mice, give the deficient diet for some time. There is a protocol, and along with the deficient diet, give PT3, our HDAC3 inhibitor. Get the pups and find out, is the memory normal? Normal means till that it can it reach to that? It should not be if it gets approved, if it works, I would say it will be huge to the society, because if you see many of the means not socially economically compromised people, they get the iron tablet, vitamin tablet from during the pregnancy from the hospital, even though they don't have the enough diet. So our intention is, if PT3 can be available in hospital in long run, maybe after my death, available in the hospital, like iron tablet or folic acid or vitamin tablet they give, but they don't give efficient normal diet. They are, because of poor, they are having deficient diet. So will the pups born have normal memory? If it happens, then it's a really good work it will be. But we are about to start that project. Then question is that if it works, then it will be supplied to everyone conceiving the child. Then the pups will not have the compromised cognition, intelligence. But then the question comes that then what about the people who are having normal diet? If you take the PT3 with the normal diet, will it increase the memory or cognition, intelligence more than the normal? That protocol also we kept and want to find out. If it is more than the normal, as per our previous experiment, it is supposed to be more than the normal. But with the normal diet and the PT3, will you have the more than this normal? If it is more, how more it is? Is it significantly more, or it is just normal? A normal is also a very relative term. Okay, so it will have the normal, so that it can be prescribed to every woman, need not to find that they are nutritionally deficient or they have normal diet. Okay, and that way we can have a bigger aim in our life to serve the society, so the kids should not born with the compromised intelligence. We all might, everyone will might born take birth with the normal memory, and then the market is open. You all fight and stand yourself. That is all about my talk. Thank you all. If you have any question, I'll be happy to answer. If you have any doubt, any interaction you want to do, I'll be very happy to interact with you. I think I finished before time.
Participants may ask their queries if they are willing to. So we have a query from participants, Monica Panugat, that we will, if we are going to inhibit HDAC, then continuous gene transcription may lead to cancer. So kindly consider and clear the doubt.
What was the question? If we inhibit?
If we are going to inhibit HDAC, then continuous gene transcription may lead to cancer.
No, we have a couple of papers. We actually, from our lab only, we published the last 2-3 years at least 10 papers. HDAC3 inhibitors actually inhibit cancer. It is the other way. Actually, we published the paper first in the cancer research, and we have proved that it is HDAC3 inhibitors actually inhibit cancer, not it will lead to cancer.
Okay.
There is a gene responsible for that. It's called tumor suppressor gene. So tumor suppressor gene actually stays suppressed with the hyperacetylation. So if you inhibit the HDAC, histone deacetylase, the suppressor tumor suppressor genes expressed and work and causes tumor suppression. It is not the way it thinks, people think. Yeah.
Thank you, sir, for sharing your deep knowledge with us. It will be a great pleasure for us to listen to you again in our future events.
It will be my pleasure too, madam.
Thank you, sir.
Thank you.
There is an announcement for the participants. E-certificate will be issued to only those participants who will submit the feedback form and qualify the online assessment. The feedback form has to be filled at the end of each day till 8 p.m. The submission of feedback form shall be counted as their attendance. The online assessment shall be conducted at the end of these stages of STTP. The link for the submission of feedback form has been posted in Google Meet chat box. Kindly go through it, the link, and submit the form. The link will be informed to you all at Telegram for 1:30 p.m. session. Thank you. I once again thank you all for your active participation. Thank you.